Hey guys, welcome to CakeWalk Cambridge. I'm Aisha and you're watching chapter 17, Inheritance. So you're lucky because this chapter is one of my favorite chapters and I know a lot of kids usually struggle with this chapter. I've also gotten plenty requests and so instead of postponing the posting of this video, I decided to make it immediately and post it for you guys who have the October-November series as well. So I hope this helps you. Let's get straight into this. Okay, so for inheritance, one thing you need to know that is important is your definitions, okay? So the first definition that's super important is the definition of inheritance itself, which is the transmission of genetic information from one generation to the next. So usually I highlight the terms that give you 1 mark each for definitions, but in inheritance, usually every definition is a mark. You're supposed to know it completely end to end, okay? So if you want an explanation for transmission of genetic information from one generation to the next, that's basically a lot of people tell me that I look like my mom. And why is that? Because some of her DNA has passed on to me, right? Through inheritance. So genetic information is passed on from her generation, from her to me, and I'm from a different generation. So that's what it is. The next thing you need to know is a chromosome. So a chromosome is basically a thread of DNA made up of a string of genes. Um, so basically this definition is very important and this could come for 2 marks if you're lucky and if you know the full definition. So just remember, if you want to remember how to like the definition of a chromosome, just remember thread and string go together. So thread of DNA, string of genes together, okay? Then you have a gene, which is a length of DNA that codes for a protein, okay? And an allele is a version of a gene. Okay, so now I'm going to give you an example. So supposing so you have different genes in your body that control your different features, right? So there is a gene that controls how tall you are. So supposing the gene for that is the gene for that is a capital T. Now a version of this gene, which is an allele, could be a small t, could mean you're short. Okay, so basically these two capital T and small t are versions of each other. Okay, so they're versions of the gene that code for whether you're tall or not. So an allele is basically a version of a gene. This is an allele and this is an allele. We'll get into this later as well, but this is just an explanation. You should know all your definitions. Okay, next. Now this is background information that you are expected to have, okay? So one is females. So basically the human body, humans have 23 pairs of chromosomes in every cell. Okay, so every body cell has 23 pairs of chromosomes, okay? Um, Now, and as we did, a chromosome is a thread of DNA made up of a string of genes. So when you're learning this, every time you read the word chromosome, say the definition in your head. So you get, you're well versed with it. So basically, we're only now looking at body cells. So body cells have 23 pairs of chromosomes. So if a scientist takes out these chromosomes from your body cell, these 23 pairs, and arranges them according to their different lengths and sizes and what they code for, okay, the result of this arrangement is called a karyotype. You do not need to know this. It's not really there in any paper definition, but this is just a technical term that you could use. So now, in this karyotype, you'll have one pair of chromosomes, which is just sex chromosomes, okay? So females have two X chromosomes that are basically like this. I'm going to draw it out for you. So they look like this, two long big ones. Whereas males have one X chromosome and one Y chromosome. Okay, so like this could be like this, but like one big one and one small one. So if you are given an arrangement of 23 pairs of chromosomes and the last pair, both chromosomes are the same size and they're big, you know it's a female's body cell. And if you're given 23 chromosomes with this kind of arrangement at the end, then you know it's a male's body cell. Okay, so now a way to remember which one is males and which one is females, just remember the word boy. Okay, boy has a Y in it, so the sex chromosomes of a boy are XY. Okay, and girls don't have a Y in it, so they're simply XX. So I know this is actually really easy to remember, But there was like some kids in my class who found this hard to remember as well. So I'd say use this technique. Boy has a Y, so boys XY, girls XX. Okay, and you should know this at like at the tip of your fingers. Okay. Yeah, so now this shows you how your sex is determined. Okay, so basically this whole diagram is called a genetic cross diagram. Okay, so all females have XX chromosomes, right? And all males have XY chromosomes. So here we're going to see if these two are interbreeded, or like if sexual intercourse takes place, which results in pregnancy, what the sex of the child could be. Okay, so now to do this, first you need to write down the chromosomes of the female and male. Then you write them individually. So one X chromosome comes here, one X chromosome comes here, one X chromosome comes here, and one Y chromosome comes here. Okay, and then after that, you need to mix and match and to find out all the possible combinations. So you can see that the different color lines indicate the different combinations. So one X chromosome of the female and one X chromosome of the male could form XX, which is a female. One X chromosome of the female and one X chromosome of the male could form XX, which is another female. Then you have one X chromosome of the female and one Y chromosome of the male, which could form XY, which is a male. And you have another X chromosome of the female and a Y chromosome of the male, which could form another male XY. So now when you're doing genetic crosses, please don't cross it in between the chromosomes. You will only cross it in between the two different like the different people you're looking at, the two parents. So you will cross these two with these two, not these two like between each other. You'll never do that. And when you have two chromosomes, you'll always have four possible outcomes. Okay. So just remember when you're doing it, there have to be four outcomes. You have female, female, male, male. And now very often in your questions, they ask you to identify the percentage chances or the probability of having a female. So what you simply have to do is apply maths here. You have two females and two males. So totally the total number of offspring that could be produced is 4, out of which 2 are males. So that basically what you need to do is 2 on 4, which is basically equal to 50%. You can leave it as a fraction, decimal, or percentage, but I usually always did it in like a percentage form, and that worked for me. So whatever you're comfortable with, you can do. And these calculations are super easy, so try not to mess it up because it's really easy, okay? So yeah, you have females and males, 4 outcomes, and or if you had to find out the probability of it being a male, you'll do again 2 males out of 4 total possibilities. So 2 out of 4, which is again 50% or half. Now what is important for you to remember is that females have XX and males have XY, and Y is the only different chromosome. So if you have a Y chromosome, you're a male. So basically, typically, essentially, yeah, the males have the chromosome that determine the sex of the child because females only have X and X, right? So basically, when you're crossing a male with a female, Y is the only difference. So that is why males carry the chromosome that determines the sex of a child. Okay. Okay, but before we go on, I just want to make it very clear. If there was a question once, an MCQ that said, Mr. and Mrs. Brown have had 3 children and all 3 of them have been girls, what is the probability that their next child will also be a girl? Now, one thing you need to remember, the amount of children you had previously is not going to determine the future or the present. Your chromosomes solely determine it. And we have already seen the genetic cross diagram in the previous slide. You could have taken a screenshot of that as well. So basically, the probability of having a female is always going to be 50%, and the probability of having a male is always going to be 50%. Okay, because we've already done the whole genetic cross and it remains the same. If it's your 7th child, 4th child, 3rd child, doesn't matter. So that's a googly, that's a trick question. Your answer is always 50%. Don't make the mistake that I did. Okay, now we're going to look at bases, pairing, and transcription, okay, which is really easy, don't worry. So in chapter 4, we saw what a nucleotide was. So a nucleotide is basically a 5-carbon sugar. Now, what is a 5-carbon sugar? It is basically a carbohydrate which has 5 carbon atoms. That's it. You don't have to get into the knowledge and depth of it. You'll do this in AS levels or IB. So a nucleotide is a 5-carbon sugar plus a phosphate group, which means it has a molecule of phosphor with oxygen and an organic base. Okay, now from all of these three terms, carbon, sugar, phosphate group, organic base, we're going to focus on organic base. Now, an organic base could be either one of A, T, G, or C. Okay, so now what do these stand for? A is adenine, T is thymine, G is guanine, and C is cytosine. You do not need to know the full forms of these at IGCSE, and you're really lucky. You just have to remember ATGC. Okay, and it's not really hard to remember. Just remember it as C tag. So CTAG. Okay, that's it. Um, and now basically three of these organic bases form one amino acid. So basically one base is found on one nucleotide, right? So supposing I have this combination CAG, TCA, GGA. This is a random combination of organic bases, okay, which is basically cytosine, adenine, guanine, thymine, cytosine, adenine, guanine, adenine. So basically, these three form one amino acid, these three form one amino acid, and these three form one amino acid. So three bases, one amino acid. One amino acid, three bases. So if you have to do the math, this comes quite often. Like I've seen this in a few papers. If they say you have 51 amino acids, find the number of bases. All you have to do is remember three organic bases, one amino acid. So 51, just going to change the color. So 51 amino acid is going to form 51 into three organic bases. You just have to cross multiply and that will give you 153 organic bases. That's literally all you have to remember as of now. Okay? You have a nucleotide. Every nucleotide carries one base. The base could be either of A, T, G, or C. Three organic bases form one amino acid. Okay? We're moving on. Yeah. So basically the sequence, okay, just gonna erase all of that because this is copied directly from the Cambridge syllabus, this last line. So the sequence of bases in a gene is the genetic code for putting together amino acids in the correct order to make a specific protein. So now every protein has a unique arrangement of amino acids, okay? So basically, the sequence of bases in it, so basically an amino acid is determined by what? Organic bases. As you can see, one amino acid is made of three organic bases. So proteins, okay, the structure of a protein is determined by the arrangement of amino acids, and the arrangement of amino acids is determined by the organic bases. The sequence of bases, okay, is the genetic code for putting together amino acids, which basically make a protein. Just remember, protein, amino acid, bases. That's basically the order of it all, the hierarchy. This is just a lot of technical terms which is in your mark scheme. You can mug up this phrase if you want to, but I just feel it's important to understand it and then explain it. Okay, now the role of DNA. I hope you know the full form of DNA. DNA is deoxyribonucleic acid, DNA. So it basically controls cell function, okay, and it controls the production of proteins, okay. Now, three of these types of proteins are: one is enzymes, two is antibodies, and three is receptors for neurotransmitters. We have already looked at Oh, we've not done receptors. Okay, that is coming in chapter 14. Antibodies comes in chapter 10, and enzymes we've already done. Enzymes are proteins. So just remember, DNA controls the production of proteins. Okay, and proteins control cell function. For example, a protein could either be an enzyme, which is used to catalyze reactions, or an antibody, which is used for your immune system, or a receptor for a neurotransmitter, which is used in coordination and response. Okay, these three terms are extremely important. Now, how a protein is made. This is called transcription, and this process is very important. Okay. So basically, you know the structure of DNA is a double helix. Okay, so it's somewhat like this. Okay, so the double helix of DNA unwinds and one strand acts as a template. So this unwinds and it comes to this. Okay, this is how it becomes. Okay. Then after that, mRNA forms alongside and makes a chain of nucleotides with complementary bases. Okay, let's break this down. What is mRNA? mRNA is messenger RNA. RNA is ribonucleic acid. M is messenger. Remember, M is always small, RNA is big. Okay, so mRNA is basically one strand only, and it is just one strand, okay, and it forms alongside. So if this is okay, this is the double helix of the DNA. It opens up, and this strand, okay, acts as the template. Okay, we're looking at this strand. So supposing the nucleotides on this strand have bases A, T, and C. Okay, so mRNA now forms a strand alongside this. I'll draw it in blue. Okay, forms a strand over here. Okay, and it makes a chain of nucleotides with complementary bases. So basically, it makes nucleotides over here. So A aligns with a nucleotide that is made by mRNA here. Is in line with a nucleotide here, and C will be in line with a nucleotide over here. Okay, but now the question is, how do we know what the complementary bases are? Okay, how do we know which base each nucleotide is carrying? Now here there is a thing you need to know. You already know your four organic bases are A, T, G, and C. What you need to remember, A always pairs with T and G always pairs with C. Okay, so A is going, so we're going to look at T first. T will pair with A, correct? So the complementary base to T is always A, and the complementary base to C is always G. Now for A it should be T, right? But the problem with mRNA is that it does not form thymine along adenine. It forms a base called uracil, which is U. So on messenger RNA, A pairs with U. Okay, but T pairs with A. So messenger RNA doesn't make T. Just remember that. Okay, A will pair with U, T will pair with A, C will pair with G, G will pair with C. Okay, that's what you need to remember. Now this is only for messenger RNA. Only on transcription, A pairs with U. U is basically uracil. But if you are going to look at DNA, two strands of DNA, if this had to unwind without messenger RNA, and this was A, T, G, then A would pair with T on this strand, T would pair with A on this strand, and G would pair with C on this strand. It's only when messenger RNA is involved will A pair with U, T with A. So it's just that T is replaced by U on messenger RNA. That is it. Okay. So mRNA forms alongside this template and makes a chain of nucleotides with complementary bases, as we have seen over here. Now this is common in MCQ. Which of the following is true? And one option was A always pairs with T. That is your answer. A always pairs with T. Okay, as you can see here, A, T, T, A. Unless messenger RNA is involved, even with messenger RNA, T and A are together, but it's just that messenger RNA doesn't make the T base, so A pairs with U. That's it. Okay, next step. Messenger RNA then diffuses out of the nucleus through the nuclear membrane. So, as you know, DNA is comprised in the nucleus. So, all of this happens in the nucleus where the DNA unwinds, messenger RNA copies it. Then, so if this is the nucleus, I'm going to make it over here. This is the nucleus. Okay, there's a nuclear membrane which surrounds the nucleus. So, the mRNA is done with its work. So it will diffuse out of the nuclear membrane through the nuclear pore. You can see this gap between the nuclear membrane. That is the nuclear pore, and that is through which mRNA diffuses out. Okay, and what it does is it goes to the cytoplasm outside the nucleus. It attaches itself to a ribosome. Ribosome is basically the site for protein synthesis, where proteins are made. The term synthesis is the building up of larger complex molecules from smaller substances using energy. So basically ribosome is the site for protein synthesis. So now since mRNA has the code of the DNA with complementary bases, it has instructions. So it attaches itself to a ribosome and instructs the ribosome to form the chain of amino acids. As you know, amino acids are made from what? Organic bases. And mRNA has what? The order of organic bases. So since it has the order of organic bases, it can instruct the ribosome to make the same order of organic bases, okay, which will then form amino acids because three organic bases form one amino acid. Okay, and one important key point to mention in your answer is that DNA stays in the nucleus. And the specific order of amino acids is determined by the sequence of bases in DNA. Okay, you add these 2 points, you get 2 more marks in your answer. So I put them in green because they are not part of the process, but they are 2 additional points that you could add to your answer. So these 4 are usually your points. You have to include all of them. Okay. So basically, so yeah. So in papers and everything, when they ask you how protein is made, you just need to look at transcription. You mention these 4 points, and you definitely add either this first one or the second one. I used to add the first one because it's much easier, so you can do the same. Let's move on. So this is basically what transcription is. So the double helix of the DNA has opened up. This is the non-template strand. This is the template strand. The mRNA, this is mRNA, okay, and it forms alongside this strand. As you can see, it has no T, but A is with U because it doesn't like have a T. But T is always with A. So as you can see, G with C, A with T, U with A. Okay, perfectly complementary. Okay, and this is your strand of DNA. So it forms these complementary bases. So, and then it diffuses out of the nuclear membrane through the nuclear pore, attaches itself to a ribosome in the cytoplasm, instructs the ribosome to arrange the bases to make amino acids, and that makes a protein. Okay, so again, A always pairs with T. That's your answer in your MCQ if you're gonna like solve a past paper. And only during transcription, A pairs with U, C always pairs with G. Okay. Okay, now we're going to look at this. So basically, now this is something that you need to be able to explain or state. This is also taken directly from your syllabus. All body cells in an organism contain the same genes, but many genes in a particular cell are not expressed because the cell only makes the proteins it needs. So basically, you have genes for height, genes to make enzymes, genes to make antibodies. So all of these genes are contained in every cell in your body. But obviously, if you have a cell that does not need to make an enzyme, it's not going to make an enzyme. So what basically happens is that gene to make an enzyme is not expressed. That's basically the concept we need to understand, that all the genes in all cells are the same, but only some are expressed. Okay, so specific proteins are made. Okay, now again, definitions. We're going to the next part of the chapter. So a haploid nucleus is a nucleus that has a single set of unpaired chromosomes, which is basically found in gametes. Now, gametes are sex cells, okay? So your sperm and ovum, egg cells, sperm cell, egg cell, they're your gametes. A diploid cell is a nucleus containing 2 sets of chromosomes, okay, which is found in somatic cells. Somatic means body cells. So I didn't know these terms until I read the textbook. So don't feel shy to like ask questions or anything. In a diploid cell, there is a pair of each type of chromosome, and in a human diploid cell, there are 23 pairs. So we're only going to focus at humans now. Focus on humans. So a human diploid cell has 23 pairs of chromosomes. So a haploid cell will have how many chromosomes? 23 pairs, sorry. So a haploid cell will simply have 23 pairs divided by 2, which is 23 chromosomes. So now I'm going to give you a question. So supposing the diploid cell of an organism has 4 chromosomes, how many will the haploid cell have? The answer to that is simple. You just have to divide the diploid by 2 and you get 2. A way to remember what it is H is for haploid, H for half, D for diploid, D for double. So that's how you multiply by 2 for diploid, divide by 2 for haploid. Okay. So that is basically what you need to know. So sex chromosomes, sex cells, human sex cells have only 23 chromosomes, whereas human body cells have 46 chromosomes. If we look at another species, the sex cells of the species would probably be 2, and the body cells of the species would be 4. Now, why this happens, you will learn in reproduction. I don't want to complicate this chapter. Just remember this for now. Okay. Okay, now we're going on to mitosis, which is a very interesting part, very scoring. So now what is mitosis? It is a nuclear division that gives rise to genetically identical cells. Okay, this is the complete definition. It's bold. You need to know the full thing. So nuclear division means the cell splits. Okay, and the 2 cells that are formed from this one cell that split are exactly identical. That's what you need to remember. Now, what is mitosis used for? It is used for growth, repair of damaged tissues, replacement of cells, and asexual reproduction. I suggest that you know at least two of these definitions. My two definitions that I always kept in mind were growth and asexual reproduction because they're easy. But I always say two uses, very important. Now this is taken from your syllabus straight away. The exact duplication of chromosomes occurs before mitosis. I will explain that to you. So when chromosomes duplicate, they form chromatids. Okay, that is basically what happens. So if this is a chromosome, okay, it'll change to this. Okay, where this is one chromatid and this is one chromatid. So they duplicate. Okay. And what you need to also state is that, yeah, the copies of chromosomes separate, maintaining chromosome number. So if the original cell has 4 chromosomes, the two daughter cells, which is the product of mitosis, will also have 4 chromosomes each. And now why? Your definition explains that to you because it gives rise to genetically identical cells. Okay, now we're going to look at the process. Okay, so first chromosomes appear to be visible in the cell, and what happens is they appear to be shorter and thicker. Okay, that's why you can see them right before mitosis. Each chromosome is then seen to have 2 chromatids. So as I showed you, they replicate themselves and they form a chromatid each. So now what happens is because these 2 chromatids are formed from one chromosome, as you can see the ratio is 1 chromosome forms 2 chromatids, the mass doubles. Okay, so this came in the March 2020 theory paper. The mass doubles for mitosis because each chromosome is seen to have 2 chromatids. The nuclear membrane then disappears. So the chromosomes and the chromatids were held together in this nuclear membrane. So what happens is this white nuclear membrane disappears. Okay, so this happens, and each chromatid is pulled to different parts of the cell. So as you can see, this chromatid will go this way, whereas this chromatid will go this way. This chromatid will go this way, and this one will go this way. So they're pulled apart to different parts of the cell, and then a nuclear membrane forms around each one. So this comes here, this comes here. A nuclear membrane forms around this, a nuclear membrane forms around this. And the cytoplasm, which was of the original cell, divides, and you have 2 separate cells. One cell here, one cell here. This is your nucleus and this is your nucleus. So you had 2 chromosomes initially, 2 chromosomes finally. So let's look at it from a diagram perspective. Here you can see you have 2 chromosomes. The 2 chromosomes then divide to form 2 chromatids, so the mass doubles. The chromatids are pulled to separate parts of the cell. So this one, I'm going to mark it yellow. This one and this one are going to go on top, as you can see here, yellow, yellow, and we'll mark the other ones in green. This one and this one go to the bottom. So they're pulled apart, and then a different nuclear membrane forms around each one of them, as you can see. This is one nuclear membrane, this is one nuclear membrane, and the cytoplasm divides. So basically, they've not drawn the cytoplasm, they should have in this diagram. So this is your cell, okay? And so if this is your cell and this is your nucleus, this is your cytoplasm, okay? This is still your cell, it's still intact, this is still it. And now over here in this stage, this divides. The cytoplasm divides and you form a separate cell, separate 2 cells. So in mitosis, you have 2 daughter cells that are produced genetically identical, same number of chromosomes. Okay. Now what you need to understand is that mitosis occurs in stem cells. Now what are stem cells? They are unspecialized cells that divide by mitosis to produce daughter cells. Okay. And now when these daughter cells are produced, usually one daughter cell continues to undergo mitosis to produce more unspecialized stem cells, while the other daughter cell stops. The process of mitosis stops there and it becomes specialized to carry out a particular function. So that's how more cells in your body are produced. So stem cells are found in skin. If you can see, it keeps dividing and increasing. Now your next part, which is meiosis. So basically, meiosis is now a little different. Okay, it's got a bigger definition, and I'd say it's a bigger topic generally. So meiosis is the reduction division in which the chromosome number is halved from diploid to haploid, which results in genetically different cells. Okay. Now basically already you can see the first key term in the definition, reduction division. which means that the cell divides, but the number of chromosomes in the cell reduces. Division, reduction, reduction, division. And then you can see chromosome number is halved from diploid to haploid. Now, meiosis happens in sex cells and gametes, as you can see. Okay, so basically, if you had 46 chromosomes, reduction division means you have 46 divided by 2, 23 chromosomes. Now, if you look at it from technical terms perspective, 46 chromosomes in a human body is a diploid number, 23 is half, so it's your haploid number. And now, because the chromosome number is half, you have genetically different cells. That's how it works. So they are used in the production of gametes. And then this is a point you will learn later in chapter 18. Meiosis is the root cause of variation, biodiversity, and leads to better adaptation because of the formation of new combinations of maternal and paternal chromosomes. So basically, meiosis produces genetically different cells, right? So that leads to variation and biodiversity. That's how it works. So okay, so now we're going to look at meiosis. So first what happens is in your cell, the chromosomes appear and lie alongside each other. Okay, so one second. So this is your nucleus. I'm going to use green for the mother and blue for the father. You have one long chromosome of your mother and one short chromosome of your mother. And you have one long chromosome of your father and one short chromosome of your father. Okay, so what happens is homologous chromosomes lie alongside each other, which means the two of them that are similar lie alongside each other. So the two long chromosomes lie alongside each other and the two short chromosomes lie alongside each other. Then what happens is the nuclear membrane disappears. So this yellow membrane that you can see disappears. Okay. And the chromosomes are pulled apart to opposite directions of the cell. So this long green chromosome and the short blue chromosome are going to go to one part of the cell. Do it in white. Okay, so this will go here, this will go here, and this will come here, this will come here. So the long blue, short green, long green, short blue. So they'll be pulled apart to opposite directions of the cell. Then what happens is after they are pulled apart, every chromosome then has two chromatids. So this green one will form a chromatid like this, okay? And the short green one will form a chromatid like this. For the blue one, it's the same, where you have two small chromatids and two big chromatids. Then what? So that happens, okay? So there are two sets of two chromatids. And then after that, a second division takes place to separate the chromatids. So the long green chromatid, okay, as you can zoom in yellow. So the long green one comes here. One of the short blue ones comes here. One long green here comes here. One of the short comes here. Then this comes here, and then another comes here. So how many outcomes do you have? You have 1, 2, 3, 4. You have 4 daughter cells that are produced and each are different. Now, what is different in meiosis is that a step comes in the middle of this where after chromatids are formed, that is called crossing over, where genetic material from each chromosome is swapped amongst the chromosomes. Okay, it's swapped. So this is not part of your IGCSE. So even these steps are like extra information, but it's always good to know them because you feel confident. So basically crossing over happens, then the chromosomes are pulled apart, okay? So, and then a boundary forms around each set of chromosomes. So one here, one here, one here, and one here. That's called the nuclear membrane. And the cytoplasm pinches off. So it separates the cells and 4 separate cells, 4 separate daughter cells are produced, okay? So basically, you just need to remember that in meiosis, it's a reduction division. Okay, the number of chromosomes is halved. As you could see in the beginning of the first cell, if you remember, okay, it was like this. One long mother, one short mother, one long father, one short father. Okay, which was 4 chromosomes in the cell, and the final product was 4 cells. as you could see like this. But this was their chromosome arrangement, if you remember. So from 4 it went to 2. So that's a reduction division. You had 4 daughter cells that were produced. And because of crossing over, there is variation. So that results in biodiversity. Okay. So your chromosomes appear, they lie alongside each other, the nuclear membrane disappears and the chromosomes are pulled apart. The chromosomes are then seen as chromatids, which means they divide into two. And then a second division takes place to divide and separate the chromatids from each other. And the nuclear membrane forms around each of the new chromosomes. The cytoplasm divides and you have 4 daughter cells that are produced. Now this is the difference between mitosis and meiosis. So mitosis produces genetically identical cells, meiosis produces genetically different cells. Mitosis has 2 daughter cells, meiosis has 4 daughter cells. Mitosis is seen in body cells, which are somatic cells. Meiosis is used for the production of gametes. Okay. Mitosis is used in asexual reproduction, as in bacteria, and meiosis is seen in sexual reproduction, as in humans. Mitosis, okay, I think there was a mistake. Okay, mitosis results in the diploid number of cells. There's a mistake here. Mitosis results in the diploid number of cells, whereas meiosis results in the haploid number of chromosomes. Sorry, mitosis results in the diploid number of chromosomes, whereas meiosis results in haploid. Why haploid? Because it's a reduction division. It divides the diploid number to a haploid number. As you saw in that previous slide where I showed you 4 chromosomes, it then became 2 chromosomes. Okay, so it's a haploid number of chromosomes. Okay, now more definitions that you need to know. Genotype is the genetic makeup of an organism in terms of the alleles present. Okay, so this is a genotype. Okay, so let's say these are the alleles. So basically, this is a, okay. Let's say B and B are the genes that code for the color of a mouse. Now, this is the gene that codes for whether it's black or not. Capital B is black, small b is brown. Let's just say that's the version. Okay, so these are alleles, so they're versions of themselves, versions of the same gene. Okay, so this is the genotype, which is denoted in symbols. Then the phenotype is the observable features of an organism. So now, okay, I will explain that to you. So if a mouse is black, in words you will write the mouse is black, and that is the phenotype. If it's spotted, then in words you will write it spotted. Phenotype is in words, genotype is in symbols. Homozygous means having 2 identical alleles of a particular gene. That would be either small b, or capital B. If we're looking at this example, okay, because they have to be identical. Heterozygous means having 2 different alleles of a particular gene, as seen in this. Okay, that's an example of heterozygous. Then a dominant allele is an allele that is expressed if present. Okay, now dominant alleles, what you need to remember is that a dominant allele is always represented by a capital letter. So in this genotype B, this is dominant and this is recessive. Recessive is always denoted by a small letter, okay, lowercase. So basically a dominant allele is an allele that is expressed if present. So if capital B is black and small b is brown, if this is your code, then your mouse is going to be black because it has a dominant allele and that dominant allele is always expressed, okay? Or if you have capital B and capital B, there's no other allele, it's homozygous, right? And when it is homozygous and has 2 capital letters, it is called homozygous dominant, okay? That's going to be B. So regardless of whether the recessive allele is present, the dominant allele will be expressed. That's why it's called dominant. And the recessive allele is only expressed if there is no dominant allele. So in this case, your mouse is never going to be brown. But if you have small b, your mouse will be brown. If you have small b, big B, your mouse will still be black because it has a dominant allele present. Okay, recessive alleles, you can think of it as dominant being stronger, recessive being weaker. So the stronger will obviously overpower the weaker. So the dominant allele will be expressed if present. The recessive allele will be expressed only if the dominant allele is not present. Okay, so if 2 identical homozygous individuals breed together, it's called pure breeding. So if you have a brown mouse that is small b that pairs with another brown mouse that has the genotype small b, it's called pure breeding. Because all the offspring it will be 100% brown. But if you have big B and big B, then it will again be 100% pure breeding. But if you have big B and small b, it will not be pure breeding because they're not identically homozygous. This is heterozygous, correct? So that is basically all your differences. Moving on. Okay. Now we're going to look at a pedigree diagram. So now we're just going to look at the different types of diagrams that you have. So one is your pedigree diagram. This is actually really easy. I'm just going to explain it to you. So you'll basically have a key in your exam. A box is usually for a male and a circle is for a female. When it's shaded, it's affected. So if they, okay, now supposing, let's just think of an example. So supposing the affected male, okay, has the alleles small s. Okay, S is actually a bad example because small and big look the same. Supposing it has it as small a. Okay, so an unaffected male would be big A, small a, or big A, big A. So usually in your pedigree diagrams, you have three case scenarios. One is affected, where the alleles are completely recessive. Okay. One is unaffected, where you have completely dominant unaffected alleles, and the third is a carrier, where you have the recessive allele. That is seen in here. Okay, one big, one small. Then you have an affected female, which would be small a, unaffected female big A, and a carrier is usually denoted with half of it shaded. That will be small a, big A. Now another thing, okay, fine. Let's focus on this first. So here, let's look at this diagram. 1 is an affected male, as you can see. 2 is an unaffected female. Okay. So when they when they breed together, they have 2 children. Both one is a male, one is a female. Both are unaffected, which means the recessive allele never got passed on. Okay. To the 5th. Okay, wait. Which means the recessive allele could have been passed on, but because there is no 2 recessive alleles. Okay, wait. Okay. So supposing 2 is a and 1 is small a. Okay. Now the possible options for 4 and 5 are capital A and small a only. You can do the test yourself in a Punnett square, which I will show you next. Okay, so you have a capital A and a small a. Okay, but because the dominant allele is present, as we learned, the dominant allele's effect is always going to be seen. Okay, always expressed. So because it has the capital A, none of them are going to be affected because they have the dominant allele. Correct? Now over here you see we have an unaffected male. So now there are 2 possible combinations. It could either have this combination, capital A, small a, or capital A, capital A. Correct? Now, how do we know whether 3, the genotype of 3 is capital A, small a, or capital A, capital A? Okay, we look at their children. Now, the fact that they have 2 children that are affected means that there was no dominant allele present in 9 and no dominant allele present in 11. Correct? So now for there to be, so there's already one recessive allele from the mother that we've already seen. That means there had to have been a recessive allele from the father because as I showed you in the genetic test cross, the crossing over happens between the mother and the father. So that means they would have had to inherit one recessive allele from the father, which means the father has a recessive allele. But the father is unaffected, which means that the dominant allele was present in the father. And so that is why the genotype of the father would be capital A, small a, which makes him a carrier of the disease. That was basically it. I hope you understood this part. If you have any questions, you can email me. My email is on the YouTube channel. So yeah, I hope I kind of made this easier for you guys. Next is your genetic diagram. I've already shown this to you guys. So first you need to always write the parents. Okay. So you have supposing the genotype of one parent is capital D, small d, and the genotype of the other one is small d, small d. So this is dominant, this is recessive. Why? Because this has a capital allele, this has only two lowercase alleles. Then you'll rewrite the alleles with separate letters. Then you'll cross them over, seen as these colors, and you will write down all the combinations together. And underneath them, you need to write the phenotype. So supposing capital D means dark, okay, and small d means light. So the phenotype of this is dark, the phenotype of this is light because it has no dominant allele. But this has dominant, so it's dark. This has only recessive, so it's light. And if they ask you to find the percentage, the probability of the parents having a light child, again, 2 light out of 4 possible possibilities. So 2 on 4, which is 50%. That is your answer. Okay. Very often, when they give you examples of female and male, they expect you to write the sex chromosomes as well. So XD, XD for a female, it would be written like this. And for a male, it would be written as X small d, X small d. And then you rewrite it here. So this whole thing comes here, this whole thing comes here, X comes here, and Y comes here. Okay. So that is basically how it works. Then you have a Punnett square, which is basically another type of genetic cross that shows you how to find out the probability of the offspring based on the genotype. So first you have the phenotypes of parents, which is tall and dwarf. Okay. No. There's a mistake here. This would be tall and tall because it has a dominant allele. Okay, so dominant and recessive. So T and small t, capital T, small t, you write them separately. Then you make this kind of square and you mix and match. So capital T gives T, T, capital. Capital T, small t gives one capital T, one small t. So you basically put them together and then you write down the different genotypes that you obtained. You've got one capital T. You've got two of these two, which is one capital, one small, and you've got one of only small. So you write them separately, and you write their phenotypes. So this has the dominant allele, so it's tall. This also has a dominant allele, so it's tall. This only has a recessive allele, so it is dwarf. And then you find out the ratio of tall to dwarf. So you have 2 plus 1, 3, so 3 tall, and 1 dwarf, so 3 is to 1. That is your ratio. Okay. Then you have codominance, okay, which is basically seen in blood groups. So codominance is basically when your heterozygous intermediate, you both alleles are expressed, no allele masks the effect of the other. Okay. Now we need to remember a lot of things in blood groups. So one is that if you're A positive, these are the possible blood groups you could have. Okay, wait. I'm just gonna rewrite it. So the gene for which blood groups is seen is called I. So you always write it as I. Okay. So if you're A positive, your possible blood groups are IA, or IAIO. Just remember, O is always recessive. A and B are always dominant. Okay. So if you're O positive, that probably means you've inherited an O allele from each of your parents. So your parents, if your mom is A positive and dad is B positive, but you're O positive, that shows that they are heterozygous. Your mom's genotype would be IAIO. Your dad's genotype would be IBIO. And here you've got the O and O from both of them, as seen in this. Okay, so let's look at this. Yeah, okay, wait, as I was saying, for A positive, these are your two options. For B positive, you've got IB. Okay, and O is just simply OO, IO. So if your mother is A positive, these are her alleles. They'll usually give it to you on the question. So IAIO. Your father is B positive, IBIO. Then you make a Punnett square and you find out the different combinations. So IAIB, IBIO, IOIA, and IOIO. Okay, this is your O positive combination. Okay, but now if you can see over here, you have IAIB. Now both of them are dominant alleles. So what do? Which one is expressed? both of them are expressed. That's called codominance, when neither of them masks the effect of the other. Okay, so that's seen in blood groups. That's called codominance. And now very often if they ask you to work backwards, so supposing they say you have a blood group of I, okay, I give you the same example, IO, and your parents have a blood group of A positive and B positive, work out their genotypes. Now, obviously, if you've got IO, that means your mother has an O allele and your father has an O allele. But they've said your mother is A positive and father is B positive, which means your mother has A and your father has B. That's basically how you work backwards. You know which one is recessive and which one is dominant. Okay, now we've come to sex-linked characteristics. So basically a sex-linked characteristic, this definition you need to know very well. They ask it very often. It's a characteristic in which the gene responsible is located on a sex chromosome, and that makes it more common in one sex than another. So as we saw in the previous example, the gene for blood group uh, um, was on a chromosome that is labeled I. Okay, now what are your sex chromosomes? X and Y, right? X, Y. So X and Y for a male and X and X for a female. So supposing color blindness, okay, supposing the allele for color blindness is small c and normal blindness is big C. So if your mother has normal vision, she'll probably be X big C, or X big C and X small c. Now, either way, she's not going to be colorblind, right? Because she has the dominant allele present. Okay. Now, in your father, you basically in sex-linked characteristics, the Y chromosome doesn't carry anything, any gene. So if the X chromosome has a capital C, then your father will have normal vision. Whereas if your X chromosome has a small c with a Y, then your father will be colorblind. Okay. So color blindness is always more common in males because the Y chromosome doesn't carry the genes. So the probability of them inheriting the small c is quite big because they don't have a bigger capital, a dominant allele to mask the effect of the small c. So that is why if you're given a pedigree diagram with an example of a sex-linked characteristic, you'll probably see that the sex-linked characteristic is more common in males. And why is that? Because it is because no gene is present on the Y chromosome. Okay, it is located on a sex chromosome. Okay, so we're looking at the genetic diagram for the inheritance of color blindness. Okay, so for a female, okay, an unaffected female carrier. Carrier means it's heterozygous. Okay, carrier means they have the recessive allele, but it's not being expressed. So they are normal, but they have the recessive allele. That is what carrier means. So capital N means normal vision, small n means color blindness. Okay, so and as you can see, X and Y are used because it's located on sex chromosomes. So now the male has capital N means that this male is unaffected and has normal vision. This female is a carrier and has normal vision. So we'll again write down the chromosomes separately, okay? And then we cross them over and we get, and this is the product of what we get. So capital X, so XN, which has normal vision, and non-carrier. Why is it a non-carrier? Because this female would be homozygous dominant. Dominant means normal vision. Okay. Then after that, we have one small n and one big N, as you can see by these green lines here. So this means that the female is unaffected because she has the dominant allele, but she also has the recessive allele, so she's a carrier. Then we have a male which has the capital N chromosome, capital N gene, sorry, on the X chromosome, which makes him unaffected because it's the dominant, and so he's a non-carrier. So Y will not have anything. Just remember, Y won't have anything in a sex-linked characteristic. And then we have small n and Y. Now, when it's small n, okay, it means that it's recessive, which means he has color blindness, and he's a carrier because he's a carrier of the disease. So he is completely colorblind. He's affected. Okay, so that's basically how it works. Okay, so just remember for a sex-linked characteristic, it's more common in one sex than the other, and that's because because the gene responsible for the characteristic is present on the sex chromosomes. Okay, So it's more common in one sex than the other. As you can see in color blindness, there's nothing present on the Y chromosome of males. So if they have even one recessive chromosome, that's it, they're doomed, they've got the disease. So that makes them the carrier of the disease. So yeah, that was basically it. I know this is a 1 hour long video, which is super long, but you know the importance of the chapter and you know how long it is. So I hope I've been able to break this down for you. In the end, I do admit I stumbled with a few words because there are so many words like chromosomes, genes, alleles. But I hope you understood this chapter. And if you did, please like this video, share this video with your friends, save it to your playlist, subscribe to my channel, comment down below if you have any suggestions or questions, and I will surely help you. You can email me if you have any questions. Don't worry about it at all. I hope this video helped you, and yeah, all the best. Let's get through this together.